So that's how to sequence really in a, very quickly. So what part of DNA should you sequence? And as we know, there's genomic DNA, there's intergenomic DNA, and there's genes. And the genes are actually made up of different parts as well. There's exons, which are really the part that actually codes for your proteins, and there's introns, which are in between those exons within the gene. And there's a promoted region as well as what's called the 3' untranslated region. And when you get an EST, this is what people are talking about. So the cDNA, the mRNA that is processed basically has a small 5' leader chain, the exons, but not the introns, which are clipped out, and also an untranslated region on that. So when somebody says, I've sequenced a bunch of ESTs, that's what they're talking about. And so what's really missing, though, that's really important in biology, is the regulatory side. You're not sequencing the promoter when you're sequencing ESTs. And so we're missing that. So that's why genomic sequence becomes very important. If you're doing Sanger sequencing, if this is a gene model with the blue spots being the exons and the introns being the little lines in between, the ESTs tend to sequence the ends of genes, mainly the 3' end of genes, in just the way the technology works. So you're getting the ends of genes, you're not getting usually the full gene. If you do transcriptome sequencing with Illumina, which is what SolCap has done in their projects, we've come up with a method where we can look at several mRNAs, so expressed genes, across tissues where we can bulk all the RNAs from these given tissues, root, leaf, flower, and fruit. We put them all in a pot. We've normalized them such that we can have low copy number of each of the transcripts represented, so we're not resequencing the same thing over and over again. And we run it through the Illumina sequencer, and then we do an assembly. And with Illumina, we can do an assembly. With the tomato data, we actually used about 60 base pairs was the average read length that we used at the time. We did this over a year ago. And that was enough to get us an assembly on its own. We had enough overlap between those to get an assembly within the variety to do that. And then what you want to do, if you're interested in variation among varieties, you want to be able to assemble them, but the redundancy is quite important. You want to see a lot of the same reads, so you have confidence that when that this is a T, and it truly is a T. And if you see a difference, you want to see that difference more than once, so that you're confident to say, this is really a DNA marker or SNP at this position. This is real data, and we show this in all the projects we do for ESTs. If the yellow line is actually sequenced with Sanger, and that's exactly what this is. This is a project in lettuce, but we do the same thing in tomato. Remember I said the 3' end, which is actually the right side of a sequence. We're usually sequencing the 3' end of a gene when we're sequencing ESTs with Sanger. But because we've just sheared the DNA and we didn't use an enzyme that could be in the middle of this gene in this case, with Illumina, these are Illumina reads or Illumina contigs, subcontigs that are assembled together, and this is the important contig. We've seen the whole length of the gene here. So now we see from the 5' end, and if we just follow the blues, you can get a contig which represents the whole gene, which is the red line in this case. And so that's really the difference between Illumina sequencing and Sanger sequencing. You really are covering the whole gene, or the whole EST, not the whole gene, the whole EST, so the whole mRNA in this case. So to take it to the next level, what the people did, the Tomato Genome Consortium has done, is they've used a number of different technologies, but to sequence a genome, it's much more complex, obviously, because you have repeats, and the DNA is a lot more complex than what you can find in genes. So to start the whole genome, you shear your DNA in random fragments. So the technology is very similar. And you sequence these random fragments, and then you put the pieces back together. And this is really what you get. This is a cartoon, but what's important is that you can't sequence everything with any one technology, and argumentatively, you may never be able to get a perfect genome. The human genome is not perfectly sequenced yet. There's still some gaps. So areas that might have a string of Gs in areas that are very highly repeated. We can't tell apart, or we can't sequence through them. So even the human genome does look like this. It has these little gaps. Are those gaps important? Maybe, maybe not. But because we've chopped everything up, when trying to put it back together, it's not perfect. And this slide is a slide I've been using actually for over 10 years. It's anatomy. It's called the anatomy of a whole genome assembly, whole genome sequence assembly. What we're interested in as plant breeders are the SNPs, the little red dots. If the smaller lines here represent the individual reads, so these could be Illumina reads, it could be Sanger reads, from whatever technology you're using, 454 reads. So you have individual reads, and with the overlap, then you can get what's called this consensus sequence, the red line that I showed in the other slide. So those are still, you still get tens of thousands, actually more like hundreds of thousands of individual contigs or consensus sequences when you sequence a genome. That's not particularly pleasing considering you have 12 chromosomes. You really want it down to 12 linkage groups, or to 12 consensus sequences. So what we do, there's a trick called mate pairing. If we know the length of a fragment and we sequence the ends of them, we can say every time we know that the sequence of this 5' end should be within 500 bases, 2 kb, 50 kb, whatever that library is made of. And we can connect, sorry, I have it backwards. We can connect these two reads over here. So although there's a gap here, we don't know the sequence in between, we know that these two should be aligned together in the genome, or on the chromosome. So that's a way of building out on your chromosome, essentially. And the next trick, or next resource that people are using, it's called a physical map. They independently map using restriction enzymes the chromosomes. They chop up the chromosomes using restriction enzymes and put them back together. So this is an independent way of getting a physical map of the DNA. And on top of that, we throw on the genetic map, the recombination map on top of that. So if these are markers that we know that are on the genetic map, we can connect that to, and we know the sequence of those genetic markers, we can now connect those to individual, what's called scaffolds, which are a combination of these contigs. And that's so you can kind of see, you can build from a single read or a SNP, but associate that to a chromosome region that is actually genetically segregating. And so by doing this, we know that the SNP over here is actually linked to the SNP over here, even though they were from a different read through the sequencer. And what I've done, I've listed an order of basically sequence length of where you might want to use these technologies in assembling let's say this anatomy of a genome assembly. So the helicose has these short reads, so does IM torrent, so it's doing just these small reads, which really can't do anything on their own unless we have some background DNA to put them against. Illumina helps you build things. 454 helps you basically do these mate pairs and lets you build these scaffolds from a number of these contigs. And then Sanger and PacBio can also help you over here. And then this is bringing in the genetic map and the physical map. So that's basically the anatomy of a genome assembly. So when we do, somebody says, I've done a bunch of Illumina reads or a bunch of helicose reads, this is the quality of data they have. It's not this. It's not the chromosome level. So that's important to know as a plant breeder. Because these sets are cheap. These guys are really cheap to generate millions, hundreds of millions of reads in a couple of weeks. And anybody can do it. But it's only important if we get it to this level. And there's a lot of projects that are just stuck at this level. They said, no, we have all this data, we can't do anything with it. So why is the genome assembly important? And that's what I'm trying to show with the last slide. It anchors basically your reads. You can get gene function from that through annotation, because if you now have the sequence of a full gene, you can blast that against other genomes that have already been annotated, where we know the function of genes, Arabidopsis, grape, poplar, rice, where a lot more work has been done in functional genomics and tomato for now. And so the gene order, you can figure out the gene order by linking to the genetic map and the physical map. Develop new gene models for tomatoes. So the tomato gene models, there's quite a number of tomatoes genes that have no other link to all other plants. other plant genes sequenced so far. So we're just going to have to figure that out. You know, they're fruit specific, tomato specific. And of course, you can look at the alleles with the SNPs. And the coup de grace is what you really want to find, the SNP that is a functional mutation that's giving you, that difference in phenotype that you're trying to follow in your plant breeding programs. And Heather, I know, is going to talk more about that. You can actually visually see this much more detailed in what's called the Genome browser, and I'm not going to talk any more about that.